Welcome back, everyone. We're going to be talking today about meiosis, the need for meiosis, why meiosis happens. I know I haven't defined what meiosis is, but before we get into any detail, I want to explain a couple things. First of all, I'm going to be using a top-down approach explaining the big picture of what's going on, why creatures need to go through meiosis, and I'm going to work my way down and get into more detail as this screencast goes on and explain some of the nitty-gritty things that you will most likely need to know for a biology course that you're taking. So let's go ahead, and the very first thing I want to point out is I want you to imagine a problem. Let's think for a moment. Let's imagine that you have sexually reproducing cells that have the normal amount of DNA that is found in the organism itself. So let's say this sperm cell on the left and the egg cell on the right both have the normal amount of DNA that the organism normally has. Let's think about what would happen after only 2 or 3 generations. What would quickly happen is you would end up with a fertilized egg, a zygote, in the next generation that would have twice the normal amount of DNA right here. And let's imagine that that same twice the normal amount was sent off to one of its sex cells over here, and then its partner had either the normal amounts of DNA or twice the normal amount of DNA going into it. What would happen as a result? Well, their zygotes would be a fertilized egg, would have either 3 or 4 times the normal amount of DNA. And if you know something about DNA, you know it's actually quite long. Like the DNA in a human cell, if you stretched it out from end to end, would be roughly 6 feet long. It's amazing that the cell can actually package that inside its nucleus. If it had 3 or 4 times as much, there would be no way it could package that. It would just be an explosion of DNA. And if you think about life depending on this process to continue, where every generation would have an increasing amount of DNA, it quickly becomes impossible for life to continue. So this is the problem. This is the problem that sexually reproducing cells and organisms face. And so the issue is, well, how do sexually reproducing organisms overcome this issue? And that answer is going to be what this screencast is about. It's about meiosis and how actually that problem gets solved. This right here is just a visual representation of what happens. You have some cell over here that starts the process. It has 2 sets of DNA. So we say it has 2N sets of DNA, 2 full sets of DNA, you could say, one from its mom and one from its dad. If we assume that we're dealing with human cells here, we are talking about 46 chromosomes, and we call that 2N. We also call that a diploid situation, di meaning 2, ploidy having to do with the complete sets of DNA. We're talking about 2 complete sets of DNA. But the process of turning from that into a cell that has only 1 set of DNA is what we're talking about today, and that is called meiosis. And because this process works, it overcomes this fundamental problem. The problem being, how do you come up with a sexually reproducing species that mixes the DNA but doesn't increase the amount of DNA in every generation? Well, this is the solution. There's 1 set of DNA from both parents. In normal cells, in normal fertilized eggs, or in normal everyday cells, you've got 2 full sets of DNA right here. And in the sex cells, you've got 1 full set of DNA for each of the sex cells. With this solution, it takes care of the issue. But that leaves open to the question, well, how? How does a cell go from a 2N structure into a 1N structure? And that's what we're going to be talking about. And so, like I said, we're going to be taking a bird's eye view and working our way down. This is kind of a bird's eye view. You start out with this 2N cell over here. It's going to go through 1 division. This first division is called meiosis 1, and that's going to be right here. Those 2 daughter cells go through another cell division called meiosis 2, and you end up with 4 daughter cells, each of them that have 1N, one full set of DNA. So let's go ahead. I'm going to start drawing this and explaining a little bit as I go. And so after meiosis 1, you can see that there has been a division. This division has separated out what are called homologous chromosomes here. So the two blue chromosomes here are homologous. That means they code for the exact same types of traits, but they may not have the exact same alleles. In other words, this one on the left and the one on the right came originally, that DNA came originally from its own parents. One with the dark green visualized gene there and the dark blue visualized gene there could be something like dark hair and light hair. Both of them code for the trait of hair, but the green right here, this gene could code, that allele could code for, say, dark hair, and this allele over here and the dark blue could code for, say, light hair. Now both of them are coding for the trait of hair color. They both have different alleles or different versions of that gene right there. And the same goes for up here. So these are homologous chromosomes up here as well. And notice the homologous chromosomes get split. They go into different daughter cells over here. So you have one, I've drawn it as a green chromosome up top and one down below, one light blue chromosome up top and one light blue chromosome down below. That process is called meiosis 1. I will go into more detail on the next slide about what meiosis 1 is, but for now just know that there is a separation of those homologous chromosomes. Next, we do go through meiosis 2. In meiosis 2, what happens is these sister chromatids actually get pulled apart. And before I go any further, I just want to explain that this over here represents a chromosome. This also represents a chromosome over here. On the left is the duplicated version of the chromosome. On the right is a unduplicated version of the chromosome. So the chromosome on the left actually has two chromatids. The chromosome on the right has one chromatid per chromosome. It still has the same information. This duplicated form on the left has essentially an extra copy of that information attached together. It's still considered to be one chromosome. However, on the right, you've got one set of that DNA, you could say. So during meiosis 2, what happens is these sister chromatids get pulled apart, and you end up with different sister chromatids in each sex cell. So let me show you what that looks like. And so here are the visual possibilities of what you could see with these 4 sperm cells as they have come from originally from that cell that we started with on the left. Now what's going to happen is that will join up with an egg, fertilize the egg. The egg will have the same chromosomes, but not necessarily the same alleles for those chromosomes. So the mother may have curly hair where the father has straight hair, or the mother may have hitchhiker's thumb while the father has a straight thumb. That kind of thing may happen. So I'm going to draw the mother's chromosomes now for these 2 chromosomes that are shown. And so here you can see that the mother has the exact same chromosomes that the father has. She must, or else this fertilization will not happen. The offspring will not be viable. But she could have different genes. She could have different alleles for those genes that they share, I should say. So the way this would look would be something like this. Okay, and so this is what the end result may be over here. This cell would have information passed down from, say, the original person, like let's say this is the father on the left and the mother on the right. This cell has taken down the green chromosome with the black information, the black allele right here. This is going to be passed down to his son or daughter right there. But notice this has not been passed down. The green chromosome with the purple allele over here that represents some other allele, some other version of a trait like hitchhiker's thumb or widow's peak or something like that. That has not been passed down just by sheer chance to his offspring. That chance effect, by the way, is called independent assortment. If you notice over here for the father, once again, we have passed down this, the blue chromosome with the green trait is being passed down, And we don't have anything with the blue chromosome with the blue trait being passed down to the offspring. However, from the mother's side, the zygote, the child, the fertilized egg, will get half its DNA from its mother. And so that's what you see being transferred over here. And yeah, so that's an overview of meiosis. I'm going to go into more detail and explain this in more detail, meiosis 1, and this in more detail, meiosis 2. So here we go. Okay, so at first glance, this image is a bit overwhelming, but I'm going to walk you through it step by step. It'll be fine. We're going to start right here. The reason why I drew it this way is because it is very long, and to divide this into two different scenarios. So right here, this portion is called meiosis 1, and the rest is called meiosis 2, and that's going to be also down here as well. And that's because you go through two divisions. You start with one cell here. This is one cell right here. You end up with two cells over here, and eventually you end up with 4 cells at the end of this. So the end of this process is going to have 4 cells, each with one set of DNA, and that's where we're going with this. Okay, and so the first thing to know about this very first step is not really in this whole process that we're talking about. It's before that process. It's called interphase, and there's more I could say about interphase, but basically the cell is doing its own thing here. The important thing to know is that during that S part of interphase, you're going to go from a non-duplicated version of chromosomes to a duplicated version of chromosomes like this, and that is going to be necessary for the stage right here. And so just to keep an overview of what's going on, we're going to use the same 4 steps that we used in mitosis to talk about what's going on here, because the same 4 steps apply, it just applies twice. So if you've seen this before, PMAT, if you think about, we've got these steps right here. Prophase is the first one, metaphase is the next one, anaphase is the third one, and telophase is the 4th that we're going to be talking about. So actually this turns out to be prophase 1, this turns out to be metaphase 1, this turns out to be anaphase 1, and this is telophase 1. Because it's happening in meiosis 1, it's the first division that we're going through, and then the cell is going to go, these cells, actually there are 2 of them at this point, they're going to go through another division and the same 4 steps as we've had before. Okay, and so similarly this is going to be prophase 2, metaphase 2, anaphase 2, and telophase 2. Similarly up here you've got prophase 2, metaphase 2, anaphase 2, and telophase 2. And basically very similar things to what you have learned about from mitosis are going to be happening in meiosis in these 2 divisions. There is another thing we need to talk about though, and that has to do with what's called crossing over. Okay, and so when you have homologous chromosomes get together, that's what I've drawn right here. You've got homologous chromosomes. They line up in prophase 1 and in metaphase 1 as well a little bit. But mostly prophase 1. There is a switching over that can happen. It's called crossing over. And what happens is arms of the chromosomes can get switched. They can literally cross over and separate from one chromosome and switch from one to another. This increases genetic variability. This means that genes that are linked together get separated out. So if there is another gene, say over here and over here, they get separated out and changed up. So you can see in this first example, you've only got orange with purple. In the second example up here, you've got this orange gene with now a red gene or a purple gene, representing different alleles for different things. So it just, at the end of the day, it increases genetic variability. It's called crossing over, and that's going to primarily happen in P1, in prophase 1. That's what's happening here. So just to reiterate, in prophase 1, the most important thing is that the chromosomes have coiled up. There's been crossing over that has occurred, so that increases the genetic variability, and we prepare for metaphase 2 where they line up in the center. Right now I'm showing them in prophase 1 kind of lined up in the center, but they might be, say, off to the side where the nucleus is broken down. And in the next phase, we're going to see something similar to this drawing over here. In metaphase 1, they're going to line up in the center and prepare for them to be pulled apart. Okay, yeah, so in metaphase 1, they're going to line up in the center of the cell, this tetrad, this pair of homologous chromosomes, each with 2 chromatids each. So we're talking about 4 chromatids in total. For each pair, you're going to have these line up in the center of the cell itself, and they're going to be prepared to be pulled apart. The homologous chromosomes will be pulled apart, that is. And so in anaphase 1, we can see these spindle fibers pulling these homologous chromosomes apart, this tetrad apart. Telophase 1, they're going to separate into 2 completely different nuclei. Okay, so at the end of telophase 1, you've got 2 new nuclei. The nuclear envelopes form around those cells. Cytokinesis pinches off the middle there, and you end up with 2 daughter cells that in this model are going to spread to the top and the bottom of the model. And that's the end of meiosis 1, and meiosis 2 is going to begin. Now meiosis 2 goes through the same 4 major steps that we've talked about for both mitosis and the first half of meiosis: prophase, metaphase, anaphase, and telophase. Again, we're going to call this prophase 2 here, and here prophase 2, followed by metaphase, anaphase, and telophase 2 for both scenarios. You end up with 4 cells at the end of this, and everything I've said previously applies to these stages as well. So I'm going to speed this up by just saying what you would expect to happen in anaphase happens, metaphase. They line up in metaphase, anaphase they're pulled apart. Telophase, you have new nuclei being formed. Prophase, at the very beginning, you're going to have the nuclear envelope being broken down, and you have the chromosomes thickening up and being prepared to be separated out. So with that, I'm going to draw the rest of this. Okay, and so one of the things I want to add in at the end here is that these 4 cells right here that you see marked with the purple check marks right here and the orange arrows, they are all 1N cells. That means they are cells of this type, where they're unduplicated single chromatid chromosomes, but more importantly than that, they've only got one version of each gene, you could say. Like up here, maybe we have a capital A for a gene that's located right here, and down here maybe we have a lowercase a for this gene that's being transported right here. We only have one type that's being sent off in each sperm or egg cell. This could be a sperm cell or it could be an egg cell, but you're only getting one set of DNA leaving through that sperm or the egg cell, and that solves our original problem. So this whole process, these 2 divisions, the crossing over and all of that, is designed to solve that original problem of not having too much DNA in future generations. Also, it's used to maximize this crossing over effect that you see over here, this crossing over business. That increases genetic variability, and that is what one of the things that life, that natural selection, that evolution all depends on. So I'm going to put together a second screencast most likely on applications of this. Thank you for listening. Hope you've enjoyed it.